Assalamu alaikum wa rahmatullahi wa barakatuh. I am medical student and in today's video lecture we are going to start the topic of growth and development, which is in particular development of chick. So let's get started. The first word that came into our mind when we hear the word chick is about the egg. And in nature we find 4 different types of egg. According to the basis of the amount of yolk present in the egg, there are 4 types. Some having large amount of yolk or enormous amount of yolk, some having a very negligible amount of yolk, some have the moderate and some have a few. So the first one is egg having the no yolk. Negligible amount of yolk means egg having no yolk present, and this type of egg is found in mammals. The second one is egg having a very reduced amount of yolk or very few amount of yolk is present, and this type of egg is found in amphibians, especially the class of amphibians cephalochordates. Now let's move on to the third type, which is egg having moderate amount of yolk, not too less, not too more. It is moderate, means half of the egg is covered by the yolk and half of it is free of yolk, or it is protoplasm. So this type of egg is found in frogs. And the fourth one or the last one is egg having a very little portion free of yolk, means all of the egg is covered by the yolk, and this type of egg is found in birds, and we can say in chick. So now we know that we are learning about the development of chick, and in chick we have egg having the enormous amount of yolk. Now we can say the type of egg of frog is called mesolacethal, and the type of egg of hen is called polylacethal, having enormous amount of yolk. Poly means many. So now we can learn about the type of egg in the chicken. Now we can learn about the shell membranes of the egg, how it is covered and which type of membranes are present in it. So little portion is free. It is free of yolk and it is covered by the protoplasm, living material, and this material is also called as a blastodisc or germinal disc. From this little portion embryo will grow, and I have made these multiple layers to show you the different names of these layers, how the well protected egg of the chicken is, because after it is come outside the body, so it needs a lot of protection. So this upper part, it is called as the blastodisc, which is living material. I have told you before. This dotted membrane, before dotted membrane we can say that this all will be filled by the yolk, and it is also, I've already told you before, and it is called yolk, and this dotted membrane is called as the vitelline membrane. And remember, vitelline is the non-cellular, it is non-cellular. And then we have albumin. Albumin is the egg white. Remember, albumin is the egg white. And then we have calcaneus or shell membrane, and it is the hardest layer, and it is calcium carbonate layer, made up of calcium carbonate. It provides the firm protection to the cell. Now let's come to the next topic, which is fertilization. Now fertilization is the fusion of the male and female gametes is called fertilization, or we can say the fusion of this ovum or sperm is called fertilization. Fertilization is of two types: external fertilization and internal fertilization. In chick we found internal fertilization. In chick we have internal fertilization. And in which part of fertilization occur in chick? Anterior part of oviduct. In anterior part of oviduct. And when it is coming out from the oviduct, it is covered by albumin outside the vitelline membrane and outside the albumin by the calcaneus shell membrane. All it is covered, all these layers covered when it is coming outside by the oviduct. So now we have the important point which you have to remember must, because it's asked in MCQs and also in the MCAT. So what it is? The egg which is released from the ovary, it is released in the form as secondary oocyte. It is released as secondary oocyte. What is secondary oocyte? When the division stops at the prophase 2 of the meiosis 2, and this secondary oocyte is not too large, just 3 centimeter. And now the question arises most common in our mind, that the egg we eat, it is fertilized or not? No, it is not fertilized at all. The egg which is not fertilized, it is laid out of the body but cannot develop into chick at all. Only the egg which is fertilized will develop into chick. Okay, so now let's come, before coming to the next topic, one more important point is that the egg laid out of the body after 24 hours of fertilization. How many? 24 hours, or you can say 1 day after fertilization. So next topic is incubation. Next topic is incubation, and incubation is done by two methods, either artificially or by the mother, naturally. Either artificially or naturally. Artificially, it is done by the machines, or we can say particularly in incubator. This machine will provide the required temperature to the egg for the further development, and it is naturally done by the chick. The temperature required for the development is 36 to 38 degrees Celsius. Now let's come to the next topic, which is cleavage. What is cleavage? It is nothing but the repeated mitotic division. When the word cleavage come in front of you, just a line came into mind: repeated a series of mitotic division. In mitosis, number of cells divide more and more, like we have more and more nucleus, more and more cytoplasm, but the genetic material remains the same. So we can now see that in type of the cleavage that done in the chick is restricted to the blastodisc. Why it is restricted to the blastodisc or only the portion of the cytoplasm? Because the yolk hinders the cell division. What does the yolk do? Although it provides nourishment, nutrition to the growing embryo, but it hinders the cell division. So the cell division is restricted to the only part of the blastodisc. So the terms used for the cleavage is in particularly with the chick or in amphibians, in frog. So the first one is meroblastic. The word mere means part. So only the part of egg is divided. That's why we use the term meroblastic or meroblastic cleavage. Incomplete cleavage, because whole the egg is not dividing, only the blastodisc is dividing. That's why we use the term incomplete cleavage. And the third and the last one is discoidal cleavage, which means that the cleavage is restricted to blastodisc. Why it is restricted to blastodisc? I have already told you before, because the yolk hinders the cell division. How does this cleavage happens? What happens that the cell divides? First, the cell appears in the two-cell stage. And how it appears in two-cell stage? It appears in the two-cell stage when the egg is dividing vertically. So the first cleavage is the vertical cleavage. For this understanding, I have first made a complete egg having a little blastodisc, and now I am magnifying this just blastodisc for the proper understanding and no confusion. So this that I have made drawing, it is just the blastodisc. So the first cleavage is vertical, and the second cleavage is at right angle to the first one. You have seen in the diagram that the first is vertical and the second one is at a right angle to the first one, and the third cleavage is at the horizontal. And now after this we can see that the 6 cells is formed in this figure, but usually 8 cells are formed. This figure is 2D, but when we see the real one, it is 8 cell stage, and these cells are not called as cells, they are called as blastomeres. The special name of these cells is blastomeres. And now after this 3 cleavage, the cleavage becomes so random and so rapid that the millions and millions of the cells are formed. And the stage having the large amount of blastomeres is called as morula, resembling the mulberry shape. So why the cleavage happen? What is the benefit of cleavage? What happens when the cleavage does not occur? So for this we have to know the outcome of cleavage. So what is the outcome of cleavage? The outcome of cleavage is cytoplasmic localization. Remember this term, cytoplasmic localization. I have told before that in mitosis, cytoplasm divides more and more, nucleus divides more and more, but the genetic material remains same. When there is random division in cleavage, some cell get more cytoplasm, more number of nucleus, some get less cytoplasm, less number of nucleus. So according to this, the fate of the cell is decided, that which cell will develop into GIT, or which will go to the nervous system, which will be mesoderm, which will be ectoderm. So the next figure that I have drawn is to show you the morula, that how it resembles the mulberry shape, the cell having so large amount of cells. And this stage of morula is not long lasting, it's very very short, and it is instantly changed into the next stage, which is blastula. What happens in the blastula? The cells divide into upper and lower layer. The 2 cells stage is formed, and this 2 cell stage is called blastula. The black one that I have drawn, it is called epiblast, and the next one that I have drawn with the blue, it is hypoblast. What it is? Hypoblast. And the cavity which is formed inside the cell, it is inside the blastodisc, it is called as the blastocele. What it is called? Blastocele. This stage is called blastula. It is very important, that's why I am repeating. The next stage is, before going to the next stage, let me clear some basic terms, which is Zona opaca and Zona pellucida. In physics, we have learned a lot about the opaque, at which the objects from which the light doesn't pass. So in zona opaca, it is the area at where the differentiation of the hypoblast and epiblast does not occur and light does not pass from this area. So this is called as zona opaca. And the central clear region having the cavity is called as zona pellucida, having the transparent fluid in it so light can pass through it. It is called as zona pellucida. The next diagram that I have shown you about the 3D diagram of the epiblast and hypoblast, that how these two germinal layers are on the embryo. Now next let's come to the next topic, which is gastrulation, and the embryo at this stage is called as gastrula. For the gastrulation, remember three terms with this particular sequence: cell division, what it is, cell division, cell migration, and cell rearrangement. What it is? Cell division, migration, and rearrangement. Usually we learn about the division, but the division only it's not necessary. The cell must reach its particular place. That if it is a cell of nervous system, then it must reach its particular place, and not just reach the particular place, it must be rearranged with a particular sequence. So the division, first it is division, then after division migration of cells occur, and when it is reached to its particular place, the cell will get rearranged the particular sequence. Now I have written over here that all the cells of the avian embryo come from the epiblast. How it come from the epiblast? We'll discuss it further. But what is avian embryo? It is the growing embryo. Now in our book there is a comparison with the same terms to the development of frog. Like the development of chick is a little bit compared with the development of frog. So we have to know about for the comparison of these terms a little bit about the development of frog. So that's why I've made first the blastula stage of the frog. In the chick we have epiblast and hypoblast. We have learned above. In the chick, in the frog, the cells divide into the upper and the lower layer, but the upper one is shorter cells called as the micromere, and the lower one are the large cells called as the megameres. And these two cells separated, so it is called as segmentation cavity. So now we can say the segmentation cavity or the blastocele in the chick are almost same, because here segments are formed. Upper layer is lower one, like upper layer have the small cells micromere, lower layer have the large cells megamere. Segments are formed, so it is called as segmentation cavity. Next stage of the gastrula, the next cell stage, I have showed that which cells will divide more and more, either micromeres or the megameres. Remember, if the micromeres divide more and more in this diagram, then in the chicks, epiblast will divide more and more. And if the megameres divide more and more, then in chicks, hypoblast will divide more and more. So we have see from this diagram that the micromeres divide more and more. So when the micromeres are dividing more and more, it pushes the cavity inside, and what happens when the cavity is pushed inside? A little dent type is formed, and this is called dorsal lip of the blastopore of frog. In this diagram, I have also shown that how this epiblast is dividing continuously, that it takes place of the position of the entire hypoblast, or in respect to the frog, how the micromeres are dividing more and more, it takes the position of all the megameres. And this is what it is called blastopore, a dorsal lip of the blastopore of the frog. Now come back again to the check for the comparison. Before comparison, I am now making again the epiblast and hypoblast to show you how the epiblast is dividing more and more. This upper layer is epiblast and this lower one, blue layer, it is called hypoblast. And you can see that the upper portion is completely divided by the epiblast, and slowly and gradually it will take place of the hypoblast as well. That's why I have told you above that all the cells from the avian embryo come from the epiblast. Now what happened that the center part of the epiblast cells dissolve or they start disappearing, forming a streak-like area, and when it is formed, like you can say that the gap is formed, and when the gap is formed, outside there is a cluster of cells, and this cell will start moving inside this gap and come towards the hypoblast. When they are moving inside the cell, so it will form the streak-like area here, and this is called as primitive streak. You can see here I have shown from the diagram that the cells from both the sides coming from downward. So in the dorsal lip of the blastopore of the frog, cavity is moving inside, and in the primitive streak cells are moving inside, so they are both are functionally equivalent. So we can now say that the dorsal lip of the blastopore frog is functionally equivalent to the primitive streak in the chick. Now let's come back again to the frog, and I have shown you that how much it is pushed inside and almost another cavity is formed. And when just these upper and lower layer are so near that they will combine and the secondary cavity is formed. The first or primary cavity is, remember, segmentation cavity in the frog and the blastocele in the chick. And this cavity or this so much inversion or so much pushing of this cavity, it is called as archenteron. It is what? Archenteron formation. And this archenteron, when the upper and the lower layer will fuse together, because they are so much near now to each other, so when they fuse together they will form the secondary gut. What it will be formed? Future gut and the secondary cavity, and the primary cavity is segmentation cavity. Now how this archenteron resembles or compares with the chick, we will discuss in the next video lecture. So if you have any question regarding any topic, you are free to ask. You can ask in the comment section, or you can DM us on the Instagram Study with Doctors. Till then, stay tuned with us and stay positive. Good luck for your future. Assalamu alaikum wa rahmatullahi wa barakatuhu.